Welcome to your channel, Crazy Info. You heard about designer cloth, designer is, designer that, but you ever heard about designer baby? Let's take brief look at designer baby, and what is it? A designer baby is a human embryo, which has been genetically modified, usually following guidelines set by the parent or scientist to produce desirable traits. This is done using various methods, such as germline engineering or preimplantation genetic diagnosis (PGD). This technology is the subject of ethical debate, bringing up the concept of genetically modified superhumans to interbreed with and eventually replace modern humans. PGD or PIGD, also known as embryo screening. It is a procedure performed on embryos prior to implantation, sometimes even on oocytes prior to fertilization. The methods of PGD help to identify and locate genetic defects in early embryos that were conceived through in vitro fertilization (IVF). The PGD procedures allow scientists to identify damaged or mutated genes associated with diseases in the oocytes or embryos by using in situ hybridization (ISH). Conversely, this technique can also help select for desirable traits by avoiding implanting embryos with genes that have SER/ES diseases. This is not a new technology. The first PGD babies, and thus also the first designer babies, were created in 1989 and born in 1990. There is already a microchip that can test a remarkable 1,500 genetic traits at once, including heart disease, seasonal affective disorder, obesity, athletic ability, hair and eye color, height, susceptibility to alcohol and nicotine addictions, lactose intolerance, and one of several genes linked to intelligence. PGD has been used primarily for medical purposes, but as the possibilities of the procedure increase, the idea of no-end medical uses has become a popular topic of debate. PGD has the ability to select an embryo based on gender preferences. The procedure is able to create a donor offspring or a savior sibling, which can assist a pre-existing offspring for medical purposes. The savior sibling is a brother or sister that is created to donate life-saving tissue to an existing child. Many countries completely prohibit PGD, including Austria, Germany, Ireland, and Switzerland. Other countries restrict PGD to medical use only, including Belgium, France, Greece, Netherlands, Italy, Norway, and the United Kingdom. In contrast, the United States federal law does not have any regulation of PGD. The other use for designer babies concerns possible uses of human germline engineering techniques, such as CRISPR, to create desired genetic traits of a child, such as disease resistance, athletic ability, and intelligence. Human genes are found within chromosomes. Humans have 23 pairs of chromosomes, 46 individual chromosomes, 23 chromosomes are inherited from the father, and 23 from the mother. Each chromosome can carry about 20,000 genes. Researchers have already connected the genes in the striped zebra fish, which control the color of the fish, to genes in humans that determine skin color. Scientists launched Human Genome Project in 1990, which have been able to pinpoint specific locations for about 12,800 specific genes within different chromosomes. Gene therapy is the use of DNA as a pharmaceutical agent to treat disease. Gene therapy was first conceptualized in 1972, with the authors urging caution before commencing gene therapy studies in humans. The techniques used in gene therapy, which are also referred to as vectors, have a method of using a healthy gene to attack and replace an infected gene. A few of the techniques are basic processing, gene doping, and viral vectors. Lee Silver has projected a dystopia in which a race of superior humans look down on those without genetic enhancements, though others have counseled against accepting this vision of the future. It has also been suggested that, if designer babies were created through genetic engineering, this could have deleterious effects on the human gene pool. Safety is a major concern when it comes to the gene editing and mitochondrial transfer. The American National Academy of Sciences and National Academy of Medicine gave qualified support to human genome editing in 2017. Once answers have been found to safety and efficiency problems, but only for serious conditions under stringent oversight. In 2018, the Nuffield Council on Bioethics issued a report which concluded that, under certain circumstances, editing of the DNA of human embryos could be acceptable. The Nuffield Council is a British independent organization that evaluates ethical questions in medicine and biology. Thank you for watching the video, guys. For more interesting videos, do subscribe to your channel, Crazy Info. See you soon in the next video.